Our next topic is to look at the phenomenon of sex-linked traits. And at first you might think that, is this non-Mendelian? But of course it is not non-Mendelian. And it simply shows that traits can be inherited on sex chromosomes in such a way that males and females can show different phenotypes. Males from the same cross can show different phenotypes from females. So let's look at this right now. And this is actually the original discovery of the first mutation in a fruit fly was the white-eyed mutation in fruit flies. And this led to the discovery of sex-linked inheritance, and in fact proved the chromosomal theory of inheritance, which was that genes are in fact carried on chromosomes. So let's look at this right now. Here is a male who has an XY chromosomal constitution for the sex chromosomes, and a female which is XX. And this is the same in mammals as well. Humans, for example, females have two X chromosomes and males have an X and a Y chromosome. Now the mechanism by which the XY chromosomal constitution causes maleness in fruit flies and the XX chromosome constitution causes femaleness in fruit flies is quite different than the way the XX chromosome constitution causes females in mammals and humans, and the XY chromosomal constitution dictates the development of males in humans. The mechanisms are different, but nevertheless the chromosomal arrangement is the same. And in this chromosomal arrangement, we refer to males as being hemizygous for a particular trait, not homozygous or heterozygous for a particular allele. And that is because the Y chromosome does not carry the same sets of genes that the X chromosome does. So a female can be homozygous or heterozygous for a particular allele. In this case, the female here is homozygous for a normal allele which causes red eyes to form, the deposition of red pigment in the eye, whereas the male here is hemizygous for an allele which causes the development of white eyes. And the white-eyed allele is, as you will see, is recessive to the red-eyed allele. But in males, since there is no X chromosome here, and the male is what we call hemizygous, the recessive mutation will show up in males if that is carried on an X chromosome. So here we have a cross whereby a white-eyed male, by virtue of being hemizygous for the white-eyed allele, and a red-eyed female, by virtue of being homozygous for the red-eyed allele, are crossed. The female will produce gametes, eggs, that contain one X chromosome, and either of those X chromosomes carries the red allele because she was homozygous. Whereas the male yields sperm, half of which are carrying X chromosome bearing the white-eyed allele, and the other half of the sperm, the other half of the gametes, the other half of the meiotic products carry a Y chromosome. So let's do our Punnett square and see how these assort. Well, the females will all end up being heterozygous for the wild-type normal red-eyed allele and the white-eyed allele. That is because they all inherit an X chromosome from their father and an X chromosome from their mother. They are XX and they are heterozygous. Since the white-eyed allele is recessive to the red-eyed allele, all the females of this cross are red-eyed. Now, all the males of this cross will end up being red-eyed as well because they will inherit a Y chromosome through the sperm from their father, and they will all inherit a red allele-bearing X chromosome from their mother. And therefore, they will be hemizygous for the red-eyed allele, and they will have red eyes. Fine. Here we have the males and females of this cross having the same phenotypes. However, if we look at this cross, the reciprocal of this cross, in which a homozygous female, which is homozygous for the white-eyed allele in the X chromosome, is crossed to a male which is hemizygous for the red-eyed allele, that is, we cross a red-eyed male by a white-eyed female, we see what will happen here. The female will produce X chromosome, or she will produce gametes which contain a single X chromosome, each bearing a white-eyed allele. The male will produce, half his gametes will carry a red-eyed allele bearing X chromosome, and the other half will carry a Y chromosome. So when we combine these, we see again in this reciprocal cross, all the females will be red-eyed because they are heterozygous for the red-eyed allele and the white-eyed allele, and the red-eyed allele is dominant over the white-eyed allele, so that's fine. But look at this, all the males here are white-eyed, and that arises by virtue of inheritance of a Y chromosome from their father, and they can only inherit a white-eyed allele bearing X chromosome from their mother. So they are hemizygous for the white-eyed allele. So we see here that the reciprocal crosses yield different results, and in this case, the females and males have different phenotypes because of the inheritance of these alleles on the X chromosome. Now, it is actually these types of experiments which led to the let's say this, which provided strong evidence for the chromosomal theory of inheritance. Because here is a case where clearly we're looking at chromosomally determined sex, so chromosomally determined gender in flies, males being XY, females being XX, and the inheritance of a trait that was carried on the X chromosome showed concomitant simultaneous inheritance along with those chromosomes, followed by virtue of following the genders of the flies that come out of this. So here was an example of clearly then a gene, alleles being born on an X chromosome, and that essentially proved the chromosomal theory of inheritance that we had talked about earlier, whereby the behavior of chromosomes during mitosis and the behavior of chromosomes during meiosis explain the segregation of Mendelian alleles, as if Mendelian alleles are carried on chromosomes, which of course we know they are. We know that genes are found in the DNA of chromosomes. So this is a very clear demonstration then of sex-linked inheritance, and that applies across species. So in humans, for example, there are colorblind alleles which cause green or red color blindness that are born on the X chromosome. And that is why in the human population, males can, with some frequency, be red-green colorblind, but females rarely are. And that is because males, like males in fruit flies, can be hemizygous for a colorblind allele, whereas females would have to be homozygous for the same allele, and the chance of that happening is the chance of that has a much lower probability than a male inheriting a single colorblind allele from his mother. So keep in mind that Y chromosomes are always passed from father to son, and males are hemizygous for whatever alleles are carried on their X chromosome that they inherit from their mother. Males always inherit their X chromosome from their mother. So this sets up our next topic. What we have not covered so far in our consideration of genetics is the phenomenon that that genes can be carried, that alleles of different genes can be carried on the same chromosome. So far, we've only followed monohybrid crosses, where we're looking at a single pair of alleles on a single pair of chromosomes, or we've looked at independent assortment when we've looked at the inheritance of alleles for different genes that are found on different chromosomes, in dihybrid crosses, for example. But now what we're going to do is look at the inheritance of genes that are found on the same chromosome. And having understood this sex-linked inheritance, let's look at a case of one of the first experiments that actually showed the behavior of alleles that are alleles for genes that are born on the same chromosome. So let's look at that right now. So here we are looking at two genes, the white gene and the miniature gene. And the alleles that have pluses by them mean they are normal wild type alleles that cause a normal phenotype for the fruit fly. We're looking at fruit flies again. This experiment was done in fruit flies. So here we have a female who is homozygous for a white-eyed mutation. So she has white eyes. But she has normal wings because the miniature allele is wild type. Whereas here is a male who is hemizygous for a normal white gene, a white plus gene, which means that he will have red eyes because he carries the normal allele, the white plus allele. Whereas he is hemizygous for the miniature mutation, which causes miniature wings. So here we have a red-eyed miniature-winged male versus a white-eyed normal-winged female. And what we see is that this will result in the inheritance. We're coloring the chromosomes so we can follow their inheritance easily. We have females that are heterozygous for the white allele on the X chromosome and heterozygous for the miniature alleles on the X chromosome. And they are wild type. They are completely normal because the wild type alleles of both the white gene and the miniature wing are dominant to their mutant partners, their mutant alleles. The males inherit this chromosome, this yellow chromosome, from their mother, so they inherit a white mutation-bearing allele and a miniature plus normal miniature allele from their mother, and they are hemizygous for those two alleles. So by default, they are going to be white-eyed, and they are going to be normal-winged because the males are hemizygous. So let's then cross these F1 individuals together. And for simplicity's sake here, let's just look at the male progeny that come out of this F1 cross. So all the males will inherit a Y chromosome from their father, and the males will all inherit one of these X chromosomes from their female, from their female mother. So here are two types of males that we expect from this cross. We expect half the males to inherit this yellow chromosome and therefore be hemizygous for the white mutant allele and the miniature wild type allele, and therefore we expect half the males to be white-eyed and normal-winged, and we expect the other half to inherit this blue chromosome, which bear the white plus and miniature mutant alleles, and therefore we expect the other half of the males to be hemizygous for these alleles and therefore to be red-eyed and miniature-winged. Well, when this experiment was carried out in the early 1900s by a lab at Columbia University, the Thomas Hunt Morgan Laboratory, it was found that 64% of the males fell into either this category or that category. That is, 64% of the males were either white-eyed, normal-winged, or red-eyed, miniature-winged. That would be about 32% would be wide-eyed, normal-winged, and 32% approximately would be red-eyed and miniature-winged. But 36% of these male offspring were either completely wild type or doubly mutant. That is, they were red-eyed, normal-winged, or white-eyed, miniature-winged. 36%. And keep this figure in mind, this 36%. So what was happening? Well, this was worked out by the members of the Morgan Laboratory and was shown to be due to an exchange of genetic material between homologous chromosomes and meiosis. Now, in essence, this looks like a breakage and reunion. The actual mechanism for this crossing over event is more complicated than that at the molecular level. But nevertheless, the net result is that physically, DNA is exchanged between one homologous chromatid and another homologous chromatid, such that we will produce in 36% of the cases, this event will occur. 30% of the meioses will undergo one of these chromosomal crossing over events, or what we call genetic recombination, such that what was really happening back here in these females is that in 36% of the meioses, we were getting an exchange here. 36% of the chromosomes produced by meiosis in these females that would produce this progeny class here, we had this crossing over event occurring, which yields then the following type of males, 36% of them. About 18% are going to carry the white plus allele, but now joined to the miniature plus allele. So 18% of the males will have this kind of hybrid chromosome. That would be white plus, miniature plus. That would be doubly wild type, in other words, red eyes and normal winged. And another 18% would be doubly mutant. That is, they would carry the white allele, which has now been joined to the miniature allele. And since these males are hemizygous, they are now white-eyed and miniature-winged. So this phenomenon of genetic recombination led the Morgan Lab to the hypothesis that I should say, the observation of these exceptional males, which were not initially expected, this led to the discovery of this phenomenon of genetic crossing over. And I should just say parenthetically that crossing over occurs when the chromatids, when the chromosomes have been replicated and consist of two chromatids. So we are exchanging material from one homologous sister chromatid, from one homologous chromatid to another homologous chromatid. I'm just not drawing them here. We're showing a two-strand exchange, when actually this is occurring at the four-strand replicated chromosome stage in meiosis. This led the Morgan team to investigate this further, and what they found was, and we now know that different that alleles of different genes will show different percentages of recombination. So it works out to be something like this. If we have a chromosome here and another chromosome here, and again, this would be occurring when the chromosomes are replicated, but I'm just showing single chromosomes for simplicity's sake. And we have alleles, let's say, at gene A here, and we are heterozygous for the A alleles. And on the same chromosome, we are heterozygous for, let's say, B alleles, for B gene. The frequency with which recombination will occur depends upon the distance that these two genes are located, the distance with which these genes are separated on the chromosome. So the longer the distance between genes, let's take a case where we have a heterozygote for the A gene again, but this time we're going to be heterozygous for a C gene as well. The frequency with which recombination occurs between A and C will be greater than the frequency with which recombination occurs between the A and B genes. So if that's the distance between A and B, and this is the distance between A and C, there's a higher probability that a crossover event will occur between A and C than between A and B. And this means that the frequency of recombinant chromosomes that we just talked about, that frequency will be greater when we look at recombination between the A and C genes than between the A and B genes. Think of it like this. Think of it that there's a finite probability of a crossing over event occurring for any given distance of chromosome length. And then if you have a larger distance, that probability then of an event occurring is greater than if the distance is shorter. Think of the probability of seeing a highway patrol vehicle on a freeway, on the 101 freeway, let's say. And if you drive a short stretch of that freeway, then the chance of seeing a highway patrol car you have a certain probability. But if you drive many hundreds of miles, the probability of seeing a highway patrol car greatly increases, because there's a finite probability per mile driven that you will see a highway patrol car. And therefore, the more miles driven, the higher the probability that you will see one. That is the case here as well. Given a finite probability of a crossover event along a chromosome length, given a unit per unit length of chromosome, if you have a finite probability of a crossover event occurring, then the longer the distance, the higher the probability that a crossover event will occur. It could occur here, it could occur here, it could occur here, it could occur here, it could occur here. Whereas here, there's not as much distance for one of these events to occur at. So that means that genes can be mapped. This led to the phenomenon of gene mapping by recombination. So we can use the phenomenon of recombination and the frequency of recombination to map the distances of genes on chromosomes. And the Columbia team came up with the notion that we would say that 1% recombination, if we have a recombination frequency of 1%, they would translate that into a chromosomal distance, which they called 1 centimorgan, 1/100th of a morgan. Morgan named after the head of the laboratory that this phenomenon was discovered in. And that we also call that 1 map unit. This is map unit. So 1 map unit equals 1 centimorgan, which equals 1% recombination. So in our previous example, where we have 36% recombination, the total number of chromosomes that are observed to be recombinant are 36%. We see that this would then translate into a map distance between the white and miniature genes of 36 centimorgans, which also equals 36 map units. So we are translating recombination frequency as observed by recombinant phenotypes. We are translating that into a physical distance on the chromosome that we are labeling a centimorgan or a map unit. So the white and miniature genes in our example are 36 map units apart, 36 centimorgans apart, because 36% of the chromosomes are recombinant between the white and miniature genes. If we had observed, let's say, a total of 20% recombinant males, then we would have said the white and miniature genes are 20 map units apart. But that wasn't the case. They're 36 map units apart. And so different genes, when we look at the recombination frequency between alleles of different genes, we find that they have different recombination frequencies. And that's because they are located at different distances from each other on the chromosome. Every gene has a fixed position on a chromosome, and since that is true, we can map genes relative to each other based on their recombination frequencies. So, there is cytological visible evidence for these crossing over events. These are called chiasmata, and you can see them, for example, in these locust meiosis. You can see crossing over events visibly, where you see the homologous replicated chromosomes are joined at particular points where crossing over has taken place. And at the electron microscopical level, you can see that these synaptonemal complexes represent points of crossing over, and there is a vast molecular assemblage holding these together. So this is one chromatid, and this is another chromatid, and the synaptonemal complex is occurring between them and holds the chromosomes together and allows for recombination or crossing over to occur. So keep this phenomenon of crossing over in mind, and in the next lecture, what we're going to talk about is an actual experiment. We'll look at a theoretical experiment looking at how genes can be mapped using the recombination frequency between them. And that's what we'll pick up with at the next lecture.